Now they're single sheets. Everybody get the hormone list. Get this one.
No, just never putting grades in it. I'm like, yeah, these people could actually like grade. Yeah, people do it in the years, on everything. Her friend was a grader finding stuff. Yes, and there's all kinds of nice class together. Yeah. And we saw long. It's we got an extra. I was fortunate to take that fun on my way up here, and seeing the next one.
Okay, I suppose we should get started. Question from the class. How do we think about the fight or flight response or the rest and digest? So how does that integrate with the entire nervous system? So when we're thinking about fight or flight, what happens first? You're thinking about the nervous system. On some level, we have to sense or perceive that there's a problem. Take a huge tiger, right? We look at fangs, we see teeth, eyes take that information in, it goes up what side of the nervous system? Sensory side of the nervous system. That goes into the brain. So eyes going into the brain, and that's going to decide whether this is a problem or not. So we're dealing usually with a fight or flight thing there. When we're dealing with fight or flight, we have usually pretty strong responses. So we're thinking about that tiger or whatever it is. I want to have a strong emotional response because that's going to lead to what? Probably staying alive. If I do things critically, well, you know, it's about 11 feet tall. Looks like its fangs are about 3 inches. I may or may not be able to withstand that. That kind of response, that intellectual response, is not going to work. We need to get to the emotional centers. We decide. So this is the sensory side. If we have the eye here, we got the sensory side. We got the central nervous system here. Sensory, which is the peripheral nervous system. Then it's going to send out on the motor side. Motor nerves for whatever response we're going to have on the other side. So what directions are we going to go with the motor? We decided what directions are we going to go first? What do we want? You want to be away. So what's the part of the motor neurons are going to be activated? Somatic, which leads to what? Skeletal muscles. And we're going to start moving as fast as we can. We're also, in conjunction with that, we're going to have an autonomic response, specifically on the sympathetic side. The sympathetic side, you're going to open up the blood vessels to the legs. You're going to open up the bronchi in the lungs. You're going to do everything in support of running away fast. What's the phrase? I don't have to be faster than Tiger. I just got to be faster than you. So these two are going to work together coming out the other side. All the while, the sensory are still working, right? So we have this tiger up here, and then if we run far enough, we got one that's that big, right? Not taking up our field of vision anymore. We're going to go through the eyeball again into the central nervous system, come out this time. If we're far enough away, what's going to happen now? And it start to calm down. So you've got the somatic response still to the skeletal muscle. We're going to slow things down. And we're going autonomic down the parasympathetic side. The parasympathetic side, and we're going to start to relax. Instead of shunning all the blood to our muscle, we're going to get some back in our brain. Talk about this next week with our guest lecturer. If you're in a sympathetic state during your exam, there's no way you can think. You have to get out of that sympathetic state and get back into the parasympathetic. So your eyes are not going to be dilated anymore. Your lungs are going to relax again. Your heart rate's going to go back down. We're going into the parasympathetic state, that rest and digest. That tiger is now far enough away not to be a problem. I help? Okay. I'm guessing everybody else needed this question answered as well, right? Is that a fair statement? We have one no. Anybody else? So, over time, our perception of danger might change. So I see this tiger down by the river. Over time, I realize he's an old, tired tiger and he's got no teeth left. His claws are all broken. I'm not going to react as strongly as I did maybe the first time I saw that tiger. So responses can be habituated or decided in the central nervous system and then adjusted accordingly. That help? Okay. So one of the things that we have to learn in life, if something stresses us out, we need to learn to perceive it, adjust that perception, either by getting rid of it or learning to react to it a different way. So we can change our perception over time and therefore change our responses on the motor side. Everybody with me on that? Okay, back to the endocrines. Any other questions on the nervous system? So as I tell all the dairy students, and I can't manage that in this class, but I have the first year students in the class, they have to ask me 5 questions during the semester. It's an assignment. Each of those is an assignment. If you have a question, chances are, especially in a class that's big, you're not the only one. I've done this stuff lots and lots of times. So if you have a question or are not understanding it, chances are somebody else in the class is not understanding it. Come and ask. I can make adjustments. I can clarify things. I pride myself. I can teach anybody anything, but I have to know who you are first. I've taken D students to A's as a tutor, as a grad student. But I have to find the way you think. I have to find a way that makes sense to you. And in a class this big, I'm presenting a couple different ways usually, but maybe one of them doesn't work for you. Maybe I have to find another way. So ask questions, and maybe we can take something away from this class that's helpful. Did everybody get pictures that they want? Okay, so lecture 33 coming down the home stretch. 8 more lectures to go after today. So I'll be in Marshall Hall all this afternoon from right after this class. My meeting this afternoon has been canceled. I'll be there if you have any questions or want to talk about anything or anything cleared up. We've got our 2 exams remaining. Talking to Professor Nyberg yesterday, it appears that they've scheduled her exam with you at 10:30, 10:40, and this one's right after that. How many people have a problem with that? So a few of you. So we're going to work together. If we can put them a little bit farther apart, how much are you willing to adjust to that? Would you be willing to take one of those exams and put it on Thursday? So I'm trying to figure out how far we can move things. I know Thursday is not acceptable, but if we move things a couple hours or next day, She's thinking of flipping her 2 exams. One is much easier. She usually does the last day of classes, but she's gonna put that, maybe switch to Wednesday and Monday. Switch those, so there's an opportunity there. So we're working on it. We should be able to go through, figure that out. So I'm going to try to finish up endocrinology here and move on to the liver. And that's going to get us into metabolism and hopefully understanding how the muscles work to support running and or pulling as we move through. So we left off last time with this slide. So what are the key points that I want you to take from this? One, you've got the pancreas. Pancreas is going to secrete glucagon. Do I care really where it comes from? Probably not. You can learn that they're from the alpha cells, but what do I care about? Glucagon is secreted when levels are low, and its purpose is to increase blood glucose. So blood glucose levels are low, glucagon is secreted, and its mission is to secrete more or get more glucose into the blood. There are 2 ways to do that. One is to pull it out of storage from the liver, or to create new, and we'll talk about this probably next week, create new glucose. So glucagon is going to go to the liver, tell the liver to release its glucose stores, breaking up glycogen. Everybody remember glycogen from the beginning of the semester? String of glucoses, break it up, dump it into the blood, and save it for, move that around. We also, in the liver, we're going to take 3 carbon units, stick them together, and create our 6 carbon glucose. So that's going to accelerate that liver, and we'll talk more about that probably Monday at the rate we're going. Gluconeogenesis, making new glucose from parts that are lying around. And this becomes very important when we're talking about the racing horse. A horse under load is going to generate a lot of lactate. We have to do something with that. So the liver generally takes that lactate and creates it, or turns it back into glucose. So we'll talk more about that next week. Insulin is going to be secreted when blood sugar is high. And insulin is going to put that sugar away. Take it out of the blood, put it somewhere else. So we're going to store a lot of it in skeletal muscle. We put it in there and we make muscle glycogen. So we have glucose there when we need it. So anybody here in sports, do that pasta meal the night before. Didn't like it. So the idea behind that is if you have a big pasta meal, pasta is made of starch. That means you're going to absorb a lot of glucose. That's going to come out, hit your liver, hit your pancreas. Your pancreas is going to say, well, I don't need this much blood sugar in the blood. I can put that away for later. So we're loading glycogen into the muscle to be used when we need it. So liver is less responsive to insulin. Adipose, which we'll talk about in metabolism next week, will take that extra glucose and make fat, storing energy for later. So we're putting it away, trying to save that glucose, put it in the right spot for later. So we've been through all these different organs and what they secrete. I kind of put it all together in a list. So this is the list you should have, at least something close to it. I may have some formatting differences, but this is sort of all the stuff we've covered. So what is the hormone? If there's an alternate way of saying that hormone, like just the abbreviations, follicle stimulating hormone versus FSH, luteinizing versus LH. We've got different ways to say that. We have a source and then target, function, or purpose. So collecting all that information we presented already and moving it through. So if you start to break it out by source, you have the ones that are coming out of the hypothalamus. As a group, what are these doing? They're moving to the anterior pituitary and starting a hormone cascade. So each of these hypothalamic hormones targets the anterior pituitary and releases a specific hormone from the anterior pituitary. We get to the anterior pituitary. So we see those releasing hormones, releasing different things from the anterior. So these 5 hormones are kind of mentioned in the previous slide as the effect or target or purpose. Now we take these over here and we have a set of different purposes depending on where we are in our cycle of life, whether we have a sex of male or female. We have the 2 hormones here from the posterior pituitary that are created in the hypothalamus but released from the nerve endings in the pituitary. So trying to simplify what we talked about. And then the other hormones from the various spots, the adrenal cortex, the medulla, thyroid, parathyroid, and pancreas. So a lot of information there. What are your thoughts about learning this? Corey? It's one of those things we just got to get a handle on. So there are lots of different ways to look at this. So I gave you a list. Most of you are not going to learn well with the list. But it might make a little bit more sense if we do something visual. So taking the hormones, particularly the ones with the hypothalamus and the pituitary gland, and getting that cascade figured out. How do we move from one to the other? So taking that same information and presenting it a different way. So corticotropin releasing hormone releases, among other things, ACTH. That's going to go to the adrenal cortex and help release cortisol, which is going to have its effects along the way. So you got one cascade. As we work our way across the page, we have gonadotropin releasing hormone, GnRH. That's going to stimulate FSH and LH secretion from the anterior pituitary. They're going to go to the gonads and have different effects. So one hormone releases 2, and then there's multiple effects. We eventually get progesterone, testosterone, and estrogens in different amounts depending on what our enzyme concentrations are in those gonads that we have and what state we're currently in. Progesterones, progestins. So progesterones, testosterones, estrogens. There's a lot of different ones. We're just going to go broadly here. Growth hormone releasing factor. In the horse, it's going to release equine somatotropin. Every species has a different subscript in front. So we talked a lot about, at least with cows, BST, bovine somatotropin. So has everybody seen those labels on the milk you buy in the store? No exogenous growth hormones here, no somatotropin, no bovine, no BST. So the thing about BST is that if you took it, or EST, equine somatotropin, you took it and injected it into a human being, nothing's going to happen. There's just enough differences that they're not going to find a receptor and they're not going to have an effect. They call it not being able to go up the evolutionary ladder. Human growth hormone will have an effect in a horse. The other way around is not going to happen. Horse growth hormone is not going to affect a horse. So if we're looking at growth, there's a whole lot of different tissues that are going to be affected by that growth hormone, that somatotropin. You've got the muscles are going to get bigger. You've got the bones are going to get longer. Your tendons, your cartilage is going to adjust to the weight that we're dealing with and the power that we're generating with our muscles. So a lot of different things are going to happen. We got receptors for that somatotropin, that growth hormone all over the place. TRH, this is probably the simplest one. Thyroid releasing hormone, thyroid stimulating hormone, and then we leave our thyroid, we get thyroxine, or at least hope we get thyroxine out. That's going to, more thyroxine we have, the higher our basal metabolic rate, the higher our metabolism, the faster we burn calories. So if I have a cousin that's got low thyroid output, so a little exogenous thyroxine helps keep her from falling asleep all the time. And then we have not a hormone cascade, but we got that nerve connection. It's going to release oxytocin. That's going to affect smooth muscle in a number of locations. We have vasopressin, ADH, a antidiuretic hormone. This is going to go and affect the kidney at the collecting tubule and concentrate our urine by pulling water back into the bloodstream. So is this a better way to present it? So visual makes a whole lot more sense, a lot less confusing than that list. So the question is, and we have a limited number of people here today, how would you like to be tested on this stuff? The lists of hormones. Okay, so how do you want me to prompt? Do you want me to give you a word bank and you have to fill everything in? Or do you want to go matching? Okay, so filling out that diagram. So if I give you that diagram blank and put numbers in there, you'll be able to put whatever word next to the number. Is that what you want to do?
Yes.
Okay, what about the rest of the hormones? We could do matching with like insulin and glucagon and the epinephrine and all those other ones. Okay, so matching, so I put the hormones in one column, the description I have in the list in the other column, mix them up, and then you have to match them up. Okay. So somebody said, no, I don't want to do the other hormones. Is that a general consensus? Not everybody wants to do this full list. Okay, so what has been done in the past is if you say there's 20 matches, 10 are for your grade and 10 are for bonus, extra credit. You want to do something like that?
Yeah.
Okay, so if you're willing to work on this stuff, there's bonus points to be had. Well, I can set up the grading where say there's 10 matches, right? 10 goes to get you to 100. The other 10, whichever they are, can get you above 100. So throwing that in, that's up to you to do 10, but I guess I can let you know in the test how many I need to at least to do to get full credit. Yes, sir.
Are there like 10 that are this? Those are required?
I can, when I make up the study guide this afternoon, I can address that issue. Everybody happy with that approach? Okay. Okay, that's the end for the third exam. We got some stuff here yet on modulation. We'll put that on the final exam 4. So modulation. So how is hormone release controlled? We have these cascades, but we need to adjust them in some way. We need some feedback as we move through. So the most common one is a negative feedback mechanism where we have, I'm skipping over some slides. We don't have time for the leukotrienes and all that other stuff. So skipping should be in the back here. Okay, so we're skipping over a few slides. So negative feedback mechanism where the end product of the cascade feeds back to the beginning and shuts the system down. So a classical example of that would be the TRH, TSH, thyroxine cascade. So we've got cold exposure. The brain says, hey, things are too cold. We're going to send the TRH to the anterior pituitary. That's going to release TSH, and that's going to pump up our thyroxine. We're going to get that out into the bloodstream. We're going to raise our basal metabolic rate and hopefully stay warmer. So what happens when this system fails? Does anybody know? Anybody heard of goiter? Anybody seen those pictures of goiters where your thyroid gland swells up to ridiculous amounts? So if we don't get enough iodine in our diet, where do we usually get iodine in our diet? Salt. It's been put in salt. So we as a population have enough iodine to create the thyroxine. So we need 4 iodines per hormone unit there. We create that thyroxine. That thyroxine feeds back and tells the anterior pituitary, we're okay, we don't need to produce any more. But if I don't have that iodine, I can't make thyroxine. There's no negative feedback. So what does the system keep doing? THS, or TSH. It keeps pumping in. I need more thyroxine. It keeps sending a signal from the anterior pituitary to the thyroid and stimulates that thyroid. It grows ridiculously big, but it can't work because there's no iodine. So when everything's normal, our thyroid's the normal size. If we don't have enough iodine, we can't do that negative feedback, and it just keeps pumping through the system, stimulating that thyroid until it grows ridiculously big. We put that iodine back in, it's reversible. So negative feedback, the end product shuts down the system. We've got enough, we've done the cascade, we've got enough, we can get where we need to. Positive feedback system works the opposite way, where the end product actually accelerates the cascade. The classic example of this is the estrogens at the end of our pathway here. So I love this diagram because it's got so much information in it. We start with the hypothalamus. GnRH goes to the anterior pituitary. It's going to increase both of these here. We've got down here, we've got the ovaries responding either with follicular growth with FSH, or it's going to spike to ovulation. If you guys done that yet in classes, LH spikes to lead to ovulation. So as the follicles grow, they're going to release more and more estrogens. That's going to have a positive feedback on this loop, so we get more GnRH. But we have this little offshoot here where follicular growth is going to feed back to the anterior pituitary through a hormone called inhibin and shut down FSH. So as one follicle dominates, it's going to produce estrogen and send back here more and more GnRH. We've shut down this side, but we accelerate this side. And that leads to a LH spike, which leads to ovulation, which forms our CL, our corpus luteum, and then that's going to negative feedback to shut down the system. So you got this kind of figure 8 pattern going. So I think of with our mares, you've got them going up, they ovulate, and then they wait. What are they waiting for? See if they're pregnant or not. So we establish that using 2 different hormones. We accelerate till we ovulate, and then we sit with progesterone and wait until we find out if we're pregnant or not. If we're not pregnant, the CL regresses, we lose the negative feedback, the GnRH accelerates again, and we go through another cycle. So a lot of information in this diagram. Follow the key up here, positive feedback, negative feedback. We set up, get ready for a possible pregnancy, and then we wait. As the CL regresses, we lose progesterone, we lose that negative feedback, and the cycle starts up again. The last one we'll talk about are circadian rhythms that are influenced by the time of the day. So the daily environmental cues need to be there to reset that circadian clock. We usually think about the pineal gland in the back of the head, figuring out what time of day it is. How do we get different cycles in there? So if we look at a 24-hour clock, so 6 hours, 12, 18, 24, 6, 12, 18, 24. In the dark hours, we have spikes of certain hormones. During the light hours, we have different adjustments that are being done there. So I know for me, if I've had a number of research trials and other events or just having fun in general, that about 4 in the morning, I get really chill. And that's right before I think my cortisol kicks in and starts getting my metabolism going, get that glucose going, get things running. But normally in that nighttime, probably that 2 to 4 for me is when my low cortisol is. Normally I'm under a blanket, tucked in real tight. I don't need that extra warmth, but that 4 a.m., that cortisol starts to kick in, or probably for me 5 or 6, starts to kick in and get my day going. Melatonin, helping us sleep, is going to kick in those dark hours. That's why we get sleepy at night. And then it's very helpful that when you're not doing anything else, growth hormone kicks in and helps us grow in our sleep when we're not partitioning energy to moving around or doing other things. Yeah. Stress, yep. It can be, but it also is just kind of necessary to keep things rolling. We need a certain amount of sympathetic to get out of bed in the morning. We just need a little bit. We don't need, tiger response. We just need, okay, alarm, okay, time to get to class, time to get moving. We need a little bit of kick in there. So we have, if you probably listen or does anybody here like bounce around shifts or anything like that related to shows? You're up all night watching animals, doing stuff like that. It messes with you, right? At least for a couple days. And that's because you're messing with your circadian rhythms. You're awake and you're not getting your hormone, growth hormone. You're not getting your melatonin. And your cortisol is like, well, am I supposed to activate or not? Yes. Muscle growth, it's an anabolic hormone, so it's going to keep your muscle mass up. It's also going to help partition energy to certain different things. Like when we give it to cows, they're generally full grown. but it switches their metabolism from I'm going to put the fat on my back to I'm going to put all that energy into the milk pail. So you give a shot of BST, the cow will bump 10 pounds of milk each day. So it's a significant amount of repartitioning. So once we're mature, once our long bones are set, it's going to help manage your metabolism. So growth hormone is probably a misnomer. We probably should stick with its official name, somatotropin. Any other questions? So we're looking at circadian rhythms. During the day, our body temperature goes up, our cortisol levels are going to be higher, our blood pressure is going to be higher at night. You're going to have the opposite. Again, I'm coldest probably 3 to 4 in the morning. I get that chill when I'm working early. There was a time there I was working, getting up 2:30 in the morning to do the 3 a.m. shift. That getting out of bed was difficult until I got acclimated to it and adjusted my circadian rhythms. Melatonin, growth hormone, testosterone, prolactin tend to be down during the day. They tend to increase at night for the reasons that we'll use those. So you have things that are set on a 24-hour basis that are kind of really part of who we are. And this is one of the problems with space travel. Or people up on the space station who see 16 dawns a day, right? They're circling the planet 16 times a day, trying to get them on a circadian rhythm without the cues that we normally associate, sunrise, sunset, is difficult. And if we go to another planet that's got a 12-hour day, we're going to be really messed up. So sorting that out, keeping that there. We've got these patterns that have been built on us through our evolution on this planet. So you've got negative feedback, positive feedback, circadian rhythms are all going to influence the levels of particular hormones. We don't need to be specific about which one is which. Just understand what the mechanism is. What is negative feedback? The end product feeds back and shuts it down. Positive feedback, the end product accelerates the cascade. Circadian rhythms, we respond to signals throughout the day. What about if we have all three at once? So if we look at the hormone patterns, again, going through the pathways of pregnancy, you can see that we have all three of these things happening at once. So you've got certain patterns that are going to happen. You've got the positive feedback system here. You've got the negative feedback system here, where LH is secreted in the male, testosterone spikes, that goes to the hypothalamus, shuts that GnRH cycle down. We start it again. We get spikes throughout the day of testosterone in the male, but they are way lower for the male than we have in the female on when we take about days here. So 40 days here, human female, we're spiking as we go through the cycle. Got estrogen mostly throughout the day, but then you have the spikes that are happening. LH feeds back, shuts things down. Estradiol, I think that's the one E2 is going to be positive there. And then you just have patterns throughout the day that you get spikes. So you've got all three working together to create different patterns depending on which gonads we have. So again, that feedback loop, you got positive feedback, negative feedback, that is going to influence your luteinizing hormone levels. Any questions about endocrinology? So next up, the organ we're going to talk about, or the system we're going to talk about, is the liver. So what does the liver do? Does anybody have any thoughts? What does the liver do? Does anybody have any clue? It is a filter, among other things. What else does the liver do? It's the center of metabolism. Our carbohydrates, proteins, our fats are all managed and monitored by our liver. So liver is a filter. It's the center of metabolism. So there are a number of different. Got it. Okay. So there are a number of different artificial organs out there. We can go to dialysis. We have an artificial kidney. We have, to a limited extent, artificial hearts. Pretty simple. We just need to pump the blood through, but nobody's going to even come close to doing all the things the liver does in an artificial setting. We'd probably need an entire building to do all the different reactions that a liver does. So liver is a spongy organ. We saw that with our dissection. It's right up against the diaphragm. It's going to cheat a little bit to the right side in the horse. You got the stomach sort of on one side and the liver on the other. They're both right up against the diaphragm. So looking at that, we saw that with the foal. We're right up against that diaphragm. We saw that spleen in there. We got the stomach on the other side, and the intestines, large intestines, all will fill in that body cavity. We're thinking about refreshing your memory here. We have the circulation when we're thinking about the liver. We have the portal circulation. Everything in the gut goes to the liver, then to the heart, then to the body again. So you have all the nutrients going to the liver first. Liver gets first crack at everything and decides, okay, what needs to be managed? What can I let go? How do I organize? How do I move through? How do I adjust things? So we mentioned when we talked about cardiac, there are two different capillary beds in the liver. So you have coming down here the portal system. That's going to drain all the intestines and stomach and the guts. That's going to come in here. What do we know about this blood right here? It's gone through the guts. It's now going to the liver. What do we know about that blood? Does anybody remember? What? Not oxygenated because it already got spent here, right? So it's low oxygen. What about the nutrient level? It's going to be very high because we've just absorbed all the stuff that we can from the guts. So coming in this way, we have low oxygen, high nutrition. We've got the aorta coming down here. We work our way to the hepatic artery. We got the second capillary bed. What do we know about this one? High oxygen, low nutrient. So you have those two capillary beds overlapping in the liver that are going to supply the liver with what it needs to do its job. It needs the nutrition from the gut. It needs the oxygen from the heart and lungs. So we've got the oxygen that's going to help promote the management of all those nutrients. We'll end with this slide. So, as I mentioned, the liver is central to all the major metabolic pathways. If it happens in the body, it comes through the liver. So, we've got a wide range of things. An artificial liver would be very, very tough. We have synthesis. We're creating new things. We have degradation, or you can call it filtering. We're getting rid of problems, breaking things down. We have connections and intermediary thoughts. And anything, one time we talk about the liver, we're talking about hepatic. So hepatocytes or liver cells, hepatic artery, hepatic vein draining through. So we'll start there next time. Yes. You talking May 1st and 3rd?
First and
30th, I guess, of April?
yeah.
Every day, April, June, and November. Okay, they're all the rest have 31. So 30 days, half September, April, June, and November. All of the rest have 31 except February. Have you ever heard that before? Okay, so I'm just 30 days. So the 30th and the 1st. Okay. Okay, so you want to take the next test early. So we should be able to do something Wednesday or Thursday afternoon, assuming I get the exam made like I should, and I'm supposed to have it done by like Monday so I can get it printed up and get back by Thursday, but I can print out one copy if you want to take it on Wednesday.
If it's easier to do it after, I can do that too.
It's up to you. You decide.
Whatever's easiest.
It doesn't matter to me. It'd be nice to get it out of the way and have it in the bank so I can grade things and get everybody done and turned around. So I would lean toward Thursday.
Okay.
If anything scheduled there, let's see what we got. So right now it's the first Thursday, so there should be no meetings I have to go to at 1, so probably anytime after 1 o'clock, I can be in my office and we can find a place for you to take it. Okay, so plan on there 1-ish or
Yeah, because I have econ after this. Normal.
Okay.
He doesn't cancel it.
okay, so you
Like,
should Thursday shouldn't interfere with that, right?
Uh,
Okay, so the holy hour is 1 o'clock.
Okay.
So in theory, though there are some equine classes scheduled then. Okay, so
Days.
remind me as we get closer and we'll make sure we get you scheduled for the 1st, okay? Thank you. Yep.